It would help if I unmute, huh? Happy noon hour here. We are excited to kick off our winter webinar series called Winter Water Talks with you here today. This series is brought to you by the Water Action Volunteers Program at UW Extension and also the Citizen Lake Monitoring Network at Extension Lakes, hosted at UW Stevens Point. I am joined here today by my co-hosts Paul Skowinski, Katie Bradford, and Steph Garcia. They're going to be helping me moderate the webinar today. And before we get started, just wanted to show you the final lineup for our series this winter, although it's starting to feel like spring today, finally, huh? So here we are today, 211 bald eagles as indicators of emerging contaminants. And then in a month, on March 11th, we have land trusts and protecting land and water for our future with John Husingfeld of the Northwoods Land Trust. Before we give it over to Sean, I just want to share a few tips for participating in today's webinar. We are recording this webinar. We're going to share the link for people to watch later. Sometimes it just takes a little time to get out because it goes through accessibility checks. And we are also encouraging you to submit questions through the Q&A tab in your Zoom window. So depending on what kind of device you're on, it might be at the bottom of your screen. We are going to leave time for questions at the end of the talk. So feel free to submit a question anonymously, or you can opt to share your name there. You can also upvote other people's questions or comments if they're of interest to you. And we are definitely going to try to respond to everyone's questions nonetheless. So, without further ado, I am going to introduce the speaker today. I'm very excited for that. We have Sean Strom. He's been at the Wisconsin DNR for over 25 years. He's been serving as a toxicologist in both the wildlife management and fisheries management programs. He's studied both legacy and emerging contaminants in a wide variety of species, including bald eagles, loons, water birds, songbirds, waterfowl, fur bearers, small mammals, and fish. So today he'll be sharing a very interesting research project about looking at bald eagles to see what they can tell us about contaminants and ecosystem health here in Wisconsin. So thanks for joining, Sean, and for sharing your knowledge. We're going to pass the mic over to you.
Okay, great. Thank you very much. So I'm going to turn my camera off and I will share my screen and let me know if you can see my slides.
That looks great.
Okay, great. Okay. Yeah, so my name is Sean Strom, and I've been fish and wildlife toxicologist with the DNR for quite a while. And I've been fortunate enough to work with bald eagles pretty much my entire career. The first part of my career looking mostly at how environmental contaminants played a role in bald eagle mortality. And then about 15 years ago, I was asked to join a project looking at how bald eagles can be environmental indicators. And my primary interest was in how emerging contaminants are affecting eagle health. So, give you a little bit of background. The Wisconsin Bald Eagle Biosentinel Program, which is now referred to as the Great Lakes Eagle Health Project, began around 1990, and the original purpose was to investigate or research whether environmental contaminants played a role in the slower recovery of certain eagle populations. And the original populations of interest were along the Fox River, Green Bay, Door County area, as well as Lake Superior. And so for quite a few years, they did monitoring there, and they ultimately came up that found that for Lake Superior eagles, the recovery was a little bit slower due to simply prey availability, food availability, where Green Bay, Fox River, the slower recovery was actually most likely due to DDT exposure. And so this is kind of a larger, kind of an umbrella project where we used nestling bald eagles to trends in environmental contaminants. We go to different populations or regions on a rotating basis. We have a long laundry list of contaminants that we look at. Legacy contaminants such as metals, mercury, PCBs, organochlorine pesticides. But then one of our main focuses now are emerging contaminants, and still put PFAS in that emerging contaminant label, even though it's not really new anymore. We've been looking at PFAS and eagles since about 2011. So we collect plasma, whole blood, and feathers, and the results provide a snapshot of contaminant exposure in Wisconsin wildlife, and these results can be useful for multiple different agency programs, and I'll show you an example of that at the end. So these are the populations that we have sampled since 2011. So, as I mentioned, a heavy emphasis has been Fox River, Lower Green Bay, Lake Michigan, Wisconsin River, which is that area right down the middle of the state. These orange dots represent our Northern Highlands or our control area, and then Lake Superior and the Apostle Islands. You might say, it's like, well, what's going on here? There's nothing going on there. That area was actually covered by the National Park Service up until about 2016, and we'll be taking those areas over starting this year. So how do we go about doing this? So we use aerial surveys via airplane. Our first flight, which usually occurs in late February or early March, counts the number of occupied territories. So eagle pairs that are at a nest. And then about 6 weeks later, we do another flight that counts the number of successful nests or nests that actually have nestlings in them. We've relied on these aerial surveys literally since the project started. We have been experimenting with pretty good results using drones. And so that might be kind of a change into the future that we might be using drone technology a little bit more often to determine whether or not nests are successful. We collect blood samples from the nestlings when they're about 5 to 8 or 5 to 9 weeks of age. And the reason we target them at that kind of specific age range is kind of multifactored. One is that they're old enough to be safely handled and blood collected. Um, Two, they're not old enough where we have to worry too much about them jumping out of the nest. They fledge at about anywhere between 12 and 14 weeks of age. So getting them 5 to 8 weeks, we're not too worried that they'll jump. And lastly, any contaminants that were transferred via the egg, by this age, that is diluted out by a what we call growth dilution. And so we know at this age, everything that we observe in their blood is from the food that they eat that's brought back to the nest. So the blood plasma we collect is submitted for organic contaminants, and then whole blood and feathers are submitted for heavy metals. Now, I'm going to go show you a series of slides that uh, are taken at the vantage of the eagle nest. And the purpose, not only because they're just cool pictures, but it gives you an idea of why eagles are such good indicators over a large spatial scale. So this first nest is Kewaunee County, about a mile inland from Lake Michigan. And you can kind of see there's turkey feathers here, looks like seagulls, some scattered fish bones. And so it's pretty clear that this pair is definitely foraging in the nearshore waters of Lake Michigan. There's some probably rabbit hare in there too, so they're obviously taking advantage of being in that kind of local agricultural setting as well. Now, compared to this nest, this is a nest on Washington Island, and being on an island nest, as you can easy to understand, their diet is much more heavily fish related. Looks like there's some northern pike and maybe some whitefish in there. So with these island nests, they rely heavily on fish. Similarly, this nest was from the Apostle Islands, walleye and whitefish. And it's hard to see, but this right here is the eagle's crop, and it was extremely full. And so this particular eaglet was very well fed, as you can see by what's left was remaining in the nest. Now again, converse that this is a nest along the Wisconsin River, and so lots of river type species of fish, carp, red horse, and then if you kindly ask the nestling to move over a little bit, what we also see is that they're feeding on some crows and raccoon. And an interesting thing about eagles from these large river systems is that they eat a lot of turtles. Lots and lots of turtle shells are found in these nests. And that's very unique to these large river systems. Oftentimes on the Wisconsin River, we would find, or our climbers would find, lots of turtle shells in the nest. This is another Wisconsin River eagle nest. I think it's just kind of cool. Those are garfish there, and in the foreground, I believe that's a bowfin. So again, varied diet, but fish heavy on these very productive large river systems. Now this one is from a lake in Vilas County, so an inland lake, our control area. And what this is, if you can see my pointer, this is actually the leg of a banded common loon. And we contacted our researcher, and that loon was actually banded on that lake a few years prior. And the point of this is that in addition to the fish bones that are in the nest, it just shows you that eagles um, are an absolute apex predator. They're at the top of the food chain. And once these nestlings get to, 5, 6, 7 weeks of age, they're old enough that they're really not in danger of being preyed upon by anything. When they're really young, raccoons can maybe get into the nest if the parents aren't around. But at this age, they're able to fend for themselves to a certain degree. And so clear apex predators, top of the food chain. And so contaminants are likely extremely indicative of what's going on in their local ecosystem. And so this is just the last nest photo. And this is a nest in Oneida County, east of Rhinelander. And you can see very unique here. This walleye here was obviously caught and filleted and either tossed overboard or tossed on shore. This just goes to show you that eagles are scavengers. The walleye in the foreground, they probably caught themselves, but eagles are scavengers. They are just as happy to take a uh, roadkill skunk versus swooping down to catch fish out of a lake. The point of all these pictures is that I hope it illustrates how good eagle nestlings are in terms of being indicators of their local environment. The foraging range for a pair of adult eagles is over a mile, and so we're pretty confident that everything that we see in the blood is indicative of that local ecosystem. So how do we actually go about collecting these blood samples? And I'll kind of go through the whole process here. So you can see here our tree climber is scaling up the tree. Once he gets to the nest, he secures himself, and then he carefully places the nestlings in these specially designed bags. He lowers the bags to us on the ground via ropes. And what's on the ground, we take a bunch of standard measurements, and these include the foot pad and some bill measurements and eighth primary feather. And the purpose of these measurements is that they can be used to estimate not only the age, but the gender of the nestling. Just to give you an example, this picture here where we're measuring the foot pad, the feet of a bald eagle, when they're developing, grow faster than any other body part. And so we can use that through historical measurements that we know that if that foot pad measures 140 millimeters or greater, it is most likely to be a female. And this has been confirmed with genetic testing. So our field estimates are pretty accurate. And so that's why we take all these standardized measurements. We place a Fish and Wildlife Service band on the foot. We collect some breast feathers here for mercury analysis. And most important step here is collecting this blood sample. And we collect about 12 milliliters of blood. Once that is done, we carefully place the nestling back in the bag and we raise it back up and it's placed back in the nest. And we really only have the nestlings out of the nest for a total of maybe 15 to 20 minutes. And so kind of like to think that we run a fairly efficient operation and we get them down, sampled, and then back up to the nest as soon as we can. So now I'll go through some of the results that we've collected over the course of the last several years. Historically, this project PCBs have always been a pretty big interest. And what we have found, and it's not surprising if you're familiar with these areas, is that eaglets on the lower Fox River. And so if you're not familiar with that area, it is considered the dam and the pier downstream to the mouth of Green Bay. And so, um, not surprisingly, with the historic PCB contamination in that area, that is reflected in the blood of the eagle nestlings. And there's different toxic thresholds that are kind of debated back and forth in the literature. The one we've used is 190 parts per billion of PCBs, total PCBs. And you can see that we can still get individuals that are above that toxic threshold. Most of the other areas in the state don't approach that, but there are still some isolated spots where we do get above that threshold. And the cleanup of the Fox River was finished in 2020. Um, and so we're hoping to get back and monitor how quickly PCB levels decrease in these birds. So DDT, and the toxic breakdown product of DDT is known as DDE, and there isn't a well agreed upon threshold, so that's why I kind of have a range for in terms of toxicity. And most of the populations are below that toxic range, with the exception of Door County. And I teased out all our results from Door County. And if you're familiar with Door County, there has been a large cherry and apple orchard industry, and we think that historic use of DDE or DDT likely is reflected now in the nestlings. And even though DDT was banned 54, 55 years ago, we can still get blood levels that are still pushing that toxic range. So that's a kind of a testament of how persistent DDT can be. And I will say it took a long time for eagles to really reestablish themselves in Door County, but probably in the last 20 years, they've really taken off where the population is quite healthy now in Door County. And so, switching to blood mercury, and for the most part, mercury is not a huge concern in terms of toxic levels, with the exception of isolated spots or locations. And I kind of teased out nestlings that were collected on nests that were on river flowages or impoundments. And those we do see much higher levels of mercury. And that's not so much because it's not source driven. It's not like these areas um, have mercury sources that are dumping into them. It's just that these flowages have the water chemistry that makes whatever mercury there more bioavailable. And so for mercury to become bioavailable, you need three things. You need sulfur-reducing bacteria, you need carbon, and you need hydrogen, meaning low pH. And those three things can be common in some of these large flowages, and so that's why we see higher levels of mercury. And same thing that ON 135 is a lake in Oneida County that's considered a high mercury lake. So low pH, high dissolved organic carbon. And it just means that whatever mercury is there, it becomes more bioavailable, and thus we see higher levels in not only the eagles, but the fish that are in that lake as well. Now, switching to PFAS. The toxic endpoints of PFAS to wildlife aren't really well known, but things are kind of leaning towards endocrine toxicity, thyroid function, reproductive toxicity, and there it's a little bit more subtle, where it might be more related to nesting behavior as well as hatching success. Immunotoxicity is one that we've been particularly interested in, and I'll talk about in a little bit, among others that we don't know. Again, there's so much uh, new research coming out about PFAS and wildlife. And the hard thing about dealing with PFAS is that it's such a large class of compounds. Scientists can't even agree about how many PFAS compounds there are. Some say there's thousands, some say there's tens of thousands. And so something that's constantly changing, it's a challenge to get a handle on. And so about a year ago, we published a paper just looking at longitudinal trends of PFAS in different populations of eagles from Wisconsin. This dates back to 2011. And kind of what we have found is a kind of a very unique pattern where nestlings that are from nests along the middle and lower Wisconsin River have significantly higher PFAS levels, or I should say PFOS. So PFOS is a specific type of PFAS. It is by far the most bioaccumulative, and at least in humans, the most toxic. And so we see the highest level of PFOS in the Wisconsin River. So what we classify the middle Wisconsin River as is essentially from Rhinelander down to the dam at Prairie du Sac. And the lower Wisconsin River is essentially the lower Wisconsin Riverway, which is from the dam at Prairie du Sac downstream to the confluence of the Mississippi River. So we don't have isolated sources, but if you're familiar with this area, that is a very highly developed stretch of river, and so it's likely not just one source, but probably multiple sources. Um, and then the rest of the populations, they're relatively similar. The Great Lakes between Michigan and Superior are pretty close. And then our Northern Highlands, which we consider our control, is kind of proving that we are correct and considering it as control because we see very low levels. They're still detectable, but at a much lower level. So, as I mentioned, there's many different PFAS compounds, and these other compounds are ones that we detect most often in eagle blood. And you can see that the patterns are fairly similar, between Fox River, Lake Michigan, our control area, um, the Wisconsin River, but they're very different for this, the APIS, that's the Apostle Islands, LSSS is Lake Superior South Shore. So very unique patterns with our Lake Superior birds. And this is just another kind of a graphic way to present it. This is a graph or a figure from a paper that we're hoping to get published this spring that kind of discusses accumulation patterns in eagles. And you can see up here with the Lake Superior and Apostle Island birds, very different patterns. We don't know why. One theory is that there was a large refinery fire in Superior a few years ago, and that, this is a remnant of that. I'm a little bit skeptical about that simply because it's to label one specific event as to changing that accumulation pattern, I think is a little bit of a stretch. I think it's probably more related to just Lake Superior itself. It's extremely slow turnover rate and just how long contaminants kind of stick around in the Lake Superior ecosystem, but still lots of unanswered questions. So one thing that I've been interested since the start is, we've what I refer to as the so question, meaning, okay, we've documented what PFAS levels are in eagles. So what's the answer to the so what? What does it all mean? And so a few years ago, I was fortunate enough to start collaborating with Dr. Emily Ruse, who's there in that picture there. She is an immunologist at the Field Museum in Chicago. And the what we were interested in determining is trying to answer the questions like what impacts is PFAS exposure having on eel nestlings? And so in our 2023 and 2024 field seasons, we tried a bunch of different experiments just to see if we could see if we could find anything that's kind of worth looking into at a more detailed level. And so we looked at molecular sexing, which I had mentioned earlier, comparing our field measurements versus um, genetic DNA uh, analysis to determine gender, looking at white blood cell counts, antibodies, and as well as gene expression analysis. And I will fully state that I am not an immunologist, and so Emily was gracious enough to loan me a few of her slides for what she's been able to find or discover. And basically, what we have found is that there is a relationship between PFAS exposure and total white blood cell count. So, as you probably remember, white blood cells are cells that actively fight infection. And so, what we're seeing is eagles that have higher levels of PFAS have lower white blood cells and thus might not be as able to fight off infections. And then, and a similar nature, looking at antibody levels, and she has identified decreases in antibodies that in birds that have higher levels of PFAS. So, antibodies are proteins that are produced by specific white blood cells, and they target specific pathogens in order to, neutralize them and get them out of the way. And so, what we're thinking is that, PFAS is not killing eagles. They're not dropping dead out of the nest, but eagles with higher levels of PFAS might not be as healthy and might not be able to fight off infections as easily as eagles with lower levels of PFAS. And so this is my last data slide, and this just kind of shows total body burden of four different classes of contaminants, PFAS, PCBs, polybrominated diphenyl ethers, which were commonly used as flame retardants, but were kind of phased out maybe 20 years ago, and then DDT. And the take-home message of this graph is that by far PFAS comprises the highest body burden in terms of contaminant load. And so Eagles of Wisconsin have more PFAS than any other contaminant. And I grew up in Green Bay, and I never thought with the historic contamination of the Fox River, I never thought that PFAS would overcome PCBs, but that absolutely has happened. The data is a little bit old. It says 2011 to 2017, and the purpose is because um, we've dropped PBDEs from our analysis simply because they were at such low levels that it wasn't worth the expense. But I still just use this graphic just to show how much PFAS is an issue with our eagles. And so I'm also interested in other emerging contaminants, and whenever available, we will have them analyzed for numerous different compounds. One that I'm especially interested in are these phthalates. So phthalates are chemicals that are used in the plastic making process. They're called a plasticizer, so they make plastic more flexible. And this monobutyl phthalate is a compound that we have routinely detected in eagle blood across the state. Um, it's gotten a little bit hard to find labs that are willing to analyze for it, but that's still something that is on our wish list to continue forward as we continue this project. So what's the big picture? What does all this mean? As I mentioned earlier, our results provide a snapshot for resource managers regarding the fate of contaminants in our ecosystems. Being an apex predator, they're representative of other species within the ecosystem. This project is very popular with the public. When we're on nests that are on private land, we always invite the landowners to come out with us. They're very, very vested, very interested. This specific picture was taken on a nest at the Brown County Fairgrounds, and we probably had upwards of 20 to 30 people standing around watching, asking questions. And so it's very popular. People are very interested, not only in eagles, but what we're doing. And it just shows that eagles are valuable and accurate indicators, useful for multiple agency programs. And a specific example of this is the Wisconsin River. And so we identified um, high levels of PFAS in the Wisconsin River in 2015. A few years later, when I had fish responsibilities added to my job, um, I thought, well, we should really start looking at fish levels in the Wisconsin River. And lo and behold, and not surprising to me, we found extremely high levels of fish all along the Wisconsin River, and we probably would still wouldn't realize that if it wasn't for that initial eagle sampling. And so, for our fish program, the Wisconsin River is a significant location of interest. Like I said, pretty much every spot that we have sampled, we have had to issue PFAS-specific consumption advice for different species. So future sampling, as I mentioned, I'm collaborating with um, Dr. Gavin Dainart and Emily Ruse from UW-Sea Grant, and we were fortunate enough that we were able to get some grants. And so this current year, we will be sampling the Mississippi River, Next year, we'll be going to the St. Croix River, which is in the northwest part of the state. In 2028, we'll be back on the Wisconsin River. And then hopefully we can get back to our control area as well as resample our Great Lakes populations. So just to summarize, we detect PFAS in pretty much every single bird, regardless of the population. Um, in terms of toxic thresholds, Um, there's been some thresholds floated around for PFOS, but they're not that strong, and don't have a whole lot of confidence in them. But regardless, we do find individuals that can be above those debated thresholds. Unfortunately, thresholds for other PFAS are not well established. Hopefully, that's something that we might be able to suggest as we continue our work. We've proven that wildlife are excellent indicators of ecosystem health and change. And kind of the take-home message is that, we're exposed to the same chemicals as these eagles. We're eating the fish, we're eating the ducks, we're eating the deer. And so it is not unreasonable to think that we're not exposed to these exact same chemicals. So if you're interested in finding a little bit more about our work, you can Google Great Lakes Eagle Health Project, and it'll bring you to the project's webpage. This is our team. As I mentioned, Gavin Daynert and Emily Ruse from Sea Grant. Dan Goltz is a DNR wildlife biologist who's one of our tree climbers. Lindsay Long is our wildlife vet that's been active in the project. Josh Martinez is a regional ecologist based out of Green Bay. Brian Dewey is a research scientist. He retired a few years ago, but he liked the Eagle Project so much that I was able to convince him to come out of retirement, and so he helps us out doing field work. Mandy Camps is a former DNR employee. She currently works for Fish and Wildlife Service, but she is actually going back to get her PhD working on this Eagle project. Mandy has helped for numerous years with field work, and she was jumped at the opportunity when Gavin was looking for a PhD student that she decided to go back to school and is now kind of coming full circle and getting her PhD, doing this eagle work. And then finally, Matt Stuber. Matt Stuber is a raptor specialist with the Fish and Wildlife Service based out of Medford, Oregon, but he is a Wisconsin native, and so we fly him home a couple times a year to help climb for us. So we have a great team to work with. And other acknowledgments, Mike Meyer is the now retired research scientist with the DNR that started this project, 35 years ago. Steve Easterly is one of our biologists that helped out in the early days. Jess Lohr is our banding coordinator. And then Ryan McGann is also one of our regional ecologists that has done a lot of the survey work for us. And numerous funding sources that help us get this work done. And with that, I will take any questions and leave my contact information up for you if you need
All
it.
right. Thank you so much, Sean. Those photographs were amazing. I would very much like to hold a baby eagle if you have an opportunity coming up. We do have some questions in the Q&A. I'm going to read to you in the order they came in. So our first question is, are the tree climbers ever attacked by the adult eagles? What are mom and dad doing while you're playing
Yeah,
with the eagle babies?
yeah, great question. Get asked that a lot. The quick answer is no. And so the parents, they usually do one of two things. They'll either perch on a nearby tree or they'll just fly around way up above, and they'll talk and they'll squawk a little bit, but no, they never swoop down, they never attack. And we've both documented, and it's been well documented in the past, that once we leave the nest and leave the area, the parents are back in the nest soon after we're gone. So, they're not disturbed or anything like that. There's old wives' tale about, oh, we're gonna leave our human scent on the nestlings. Well, birds don't smell, or they have very poor senses of smell, except for condors and vultures. And so no, no, they don't smell us or anything like that. So no, the parents pretty much leave us alone. They talk and chatter a little bit, but that's about it.
All right. Do you have any ideas why the Middle Wisconsin River and the Upper Fox have higher PCB levels than the Northern Highlands?
Yes. So for Fox River, the lower Fox River or Fox River area of concern, was historically contaminated with PCBs due to paper mill contamination. And so, like I said, that's from the De Pere Dam downstream to the mouth of Green Bay. And so that remediation was completed in 2020. But historically, that's why we've seen the highest levels of PCBs. Similarly, and that middle stretch of the Wisconsin River, like say from Wausau downstream to maybe Sauk, Columbia County, there's highly industrialized, urbanized, and a fair amount of paper mills there too. And so that likely corresponded with the higher levels of PCBs in that population as well.
Next question is also about PCBs. Do the PCBs decrease in an individual eagle over time, or did they stay elevated throughout their life if contaminants are removed from the environment?
So if they're eating from the contaminated source, they will either stay steady or increase. If the source of their diet is removed, levels would likely decrease slowly. The problem with PCBs is that they're so lipophilic that they attach to fat molecules and they just don't let go. And so that's one of the bad things is that they're hard to get rid of. But they can respond, if the what they're eating is quote unquote cleaner, then likely that the levels will either stay stagnant or slowly decrease.
Okay. Do you ever test the eagles for neonicotinoids?
No, and primarily because one, getting a lab that can analyze blood plasma for neonics is a little bit tricky. And also, it's our opinion that eagles probably aren't the best indicator species for neonic contamination. Songbirds or insectivores were probably much, much better. So, there's been a lot of work with tree swallows and neonics, bats and neonics, species like that have a little bit more of a stronger connection to neonics and contaminated seeds and plants. But no, we have not tested eagles.
Right. Next on to mercury. With your
Mhm.
mercury results, will the flowages need to be treated in any way, such as through phytoremediation by the DNR or another org? Or are they
No.
isolated enough that the treatment like this could be effective?
No, no. The levels aren't high enough that it would necessitate any sort of remediation. It's just like I said, it's very localized and it's just related to water chemistry. So, for example, like Oneida and Vilas counties, where we have quite a few quote unquote high mercury lakes. There's no point sources, and you can have one lake that's a high mercury lake, but then a mile in any direction have one that's not. And so it's just local water chemistry that makes the mercury more or less bioavailable.
Okay. And I think we were getting to this with an earlier question, but just to confirm, someone's asking why there are high PFAS in the middle Wisconsin River. Was that with the paper mills that we were talking about
Well,
earlier?
we're not entirely sure. There has been the source identified up around Rhinelander, but my opinion is that that's probably not enough to result in what we see for the rest of the river. So I think it's likely multiple sources. In that middle stretch of the Wisconsin River, there's lots of industry, lots of urbanization. And so, like I said, it's probably multiple sources.
And then one of our participants is asking, we have a steady stream of questions coming in here. They're also
Sure.
fascinating too. Um, one person said they often see eagles eating roadkill deer. Is there any concern about eagles and chronic wasting disease, CWD?
That's a great question. I don't think anybody has ever looked at eagles. I know they have looked at other scavengers. I think they've looked at like maybe coyotes, and I think some corvids, maybe like crows, ravens. But I don't think anything has been firmly established or connected.
How about lead monitoring? Is that being
Yes, so we do include lead in um, our whole blood monitoring, and what we have found, and others have found too, is that lead exposure, lead toxicity, lead poisoning is much more of a concern for either adult or fledged eagles, and the reason being is that what we have seen is that lead poisoning peaks in the fall during the hunting season. So it's likely from eagles consuming either contaminated game or gut piles. And it's just that that type of contaminated food source really isn't on the landscape as much when these nestlings are being raised and subsequently sampled. So we detect lead, but it's not at the toxic level. So lead is definitely an issue with eagles. It's just a little bit of if it's a different story where it's more of a concern for adults and fledged eagles compared to these nestlings.
sure, sure. Someone is asking what can I do as an individual to help clean up the waterways PFAS problem?
Oh boy, that's a tough one. Um, you know, it's hard to say from a individual. What can individuals do? I think support programs on a larger scale that might be able to do something. There's the PFAS bills in the legislature. You can write your legislatures and say, let's get this out the door because it would provide a lot more resources to do the work that needs to be done. Things like that.
What relationships have you observed between contaminant concentrations and the number of eaglets and fledglings produced?
Sure. We don't think that there is a relationship between contaminants and the number of chicks produced in a clutch, meaning, contaminated birds are more likely to only have one chick versus two or three. Um, we have not seen any sort of connection there. Um, my opinion is that uh, the number of eggs is probably more related to resource availability. So if they're nesting in an area where there's lots of food, they may have more energy to put into extra eggs. Um, so whether or not that contaminants play a role in that is a little bit more murky.
Sure. Have the eagle populations changed over the past 2 years in various parts of Wisconsin?
In the last 2 years,
10
um,
years.
oh, 10 years. Yeah, it's probably increased. The DNR stopped doing their statewide eagle monitoring in 2019. And, the purpose was that, the eagles have recovered to a point where doing annual surveys was hard to justify. Makes our job doing this contaminants monitoring a little bit more difficult because we relied on those surveys. So had to get a little bit more creative in doing that. But we do take advantage now of citizen science and the Nest Watch program, And I believe as of a year ago, I think there's maybe been eagle nests identified in every county in the state, I think.
We love a good citizen science program. All right. What is the sample size, the number of birds tested per drainage area?
As many as we can get. So it varies. For example, this year in the Mississippi River, which is spatially, we're going all the way from the Iowa border up to Pool 3, which is on the Minnesota border, and we're hoping to get anywhere between 30 and 60 samples. And so um, it all depends on how big of an area we're looking at. If we're doing the Northern Highlands, that might be down to like say 20. And so it kind of varies with the population that we're studying in any given year.
All right. Can higher PFAS levels cause more years, like in 2022, with avian flu causing high fail rates at Wisconsin nests?
That's something that we're looking into. So yeah, so 2022 was a very bad year. That strain of avian influenza hit eagles hard. I was actually supposed to do um, so our Eagle project for funding resources, we had to put it on hiatus for a few years between 2018 and 2022. But once we started up again, it was that AI year, and we actually had to postpone it simply because it was so bad. One thing that Emily is interested in is trying to figure out, are birds that have higher PFAS levels more likely to succumb to AI? And we don't have an answer for that yet, but that's something that we're interested in.
Right. And AI being avian
Avian
influenza in
influenza.
this case. Yeah.
Yes. Yep. Yeah. Not artificial intelligence.
With their little laptops.
Yeah.
All right. We are wondering if other states are doing comparable sampling of eagles and if there are any similarities or differences in the contaminants in eagles in other parts of the United States.
So, yes, the state of Michigan has been doing this pretty much since Wisconsin started. So, Michigan started their program early 80s, early 90s. They've been fortunate enough that they've been able to do it continuously, where ours has been a little bit on and on again, off again. Um, so yes, they look at the same things that we do. In fact, I was on a meeting yesterday with staff from Michigan, where we're trying to work together on some Lake Michigan sampling. And so, yeah, so Michigan has been active. The National Park Service was very active up until 2016. And they looked at primarily like Lake Superior in those western parts of of the state that we've kind of taken over now. And we actually developed the protocols together. So we found our results are completely comparable because we're following the same protocols. And so those are the three programs that have done a lot over the last 20 to 30 years. There has been some work in the New England states, but it's been much more infrequent.
Okay. Any chances that there are plans to expand the study to rivers with higher development and contamination, like down in the Rock River?
Um, it depends on, we need enough eagle nests to make it worthwhile. So we need stretches of river where we can get, a large enough sample size to make the numbers mean something, if that makes sense. And so, no, we don't have the Rock River on our radar yet, but that doesn't mean that it wouldn't be in the future.
Okay, it looks like that is all the questions that we have here for now. Thank you so much for joining us, Sean.
My
You're getting
pleasure.
a lot of um, thanks and gratitude and the chat and the emotions that we have, the emojis, reactions that we have coming in here. So appreciate your time, appreciate all the great information, and have a great rest of your day.
You too.
To
Thanks much.
everyone.
All right. Bye.